Yes, thanks a lot. So COSSMO is a new splicing code that uses deep learning. So we know about a really long list of regulatory elements that control splicing. And the most important ones for a spliceosome to bind to a transcript and splice out the intron are that it needs to recognize the consensus motifs and the donor and acceptor ends, and recognize the branch point upstream of the acceptor motif. But there are many more cis-acting elements that also affect this process. They're called splicing regulatory elements. They can occur in the exon or the intron, and they can either inhibit or enhance the splicing. So what do we do if we have a situation where the spliceosome could choose one of multiple sites? So just having this list of regulatory elements doesn't help us very much. What we'll need to do is we'll need to integrate all these features into a computational code that can actually weigh the strengths of all these different features and tell us what the result will be. And this whole objective is very well summed up in the title of this review paper by Wang and Burge from 2008, from a parts list of regulatory elements to an integrated splicing code. And since this has been proposed, many splicing codes have been built by different groups, but we are still very far from solving that problem. And with this work, we're primarily focusing on 4 improvements to previous models. So the first one is that splicing is not always binary. Very often we have the choice between 2, 3, or more different splice sites. And so our objective is that the algorithm can make predictions for any possible number of splice sites. Secondly, we want to learn from the sequence directly. Before, people have usually built libraries of hand-engineered features, but the first problem is you need to know very well which features you want to put in that library, and better make sure that you don't miss any. And secondly, building these pipelines can really ruin your life. It's very hard and tedious and often doesn't scale well. So if you've seen Olga's talk this morning, we know that convolutional neural networks can do representation learning, where we can learn these kinds of features from sequence directly using convolutional neural nets, and this is what we're attempting here. Third, we want to model the competition between splice sites. So if you have a given splice site, its probability of binding to the spliceosome is also affected by the strength of neighboring sites. And we want to make sure that our model can control for that kind of effect. And fourth, we believe it's very important that we include background sequences, so sequences that are not splice sites in our models, in order for the model to even learn how a splice site looks different from a non-splice site. So ideally, we would want a model that can predict any possible donor being spliced to any possible acceptor. But here we're introducing a little bit more structure. So we are building 2 models. In the first model, we are conditioning on a donor site, and we consider multiple possible acceptor sites. And in the second model, we are conditioning on an acceptor site and are considering multiple alternative donors. The 2 models are exactly analogous to each other, so for the purposes of this talk, I'm just going to focus on the first case, but you can see the full details in the proceedings. So the first important step is that we need to construct a data set that we can train our model on. And we are using 2 principal data sources for this. The first one are the GenCount genome annotations, and the second one are RNA-seq reads from the GTEx consortium. So we start by picking a conditional donor site from the genome annotations, and then we look in the genome annotations and we're adding in all of the annotated acceptors that are spliced to this donor. Next, we are looking at the RNA-seq data, and we're adding in all of the de novo acceptors that we can find that are also spliced to this acceptor, subject to some filters like a certain minimum number of reads, and we need to see these sites in more than one sample. So that gives us our positive set, and we also add two different kinds of negative sites. So for the first type of negative sites, we actually want to pick negative sites that look very, very similar to real splice sites, but are nevertheless never used. And to do this, we use the max-and-scan algorithm, which predicts the splice site strength score. And we scan it along the genome, and keeping sites that have a relatively high score of greater than 3, but for which we can't find any evidence in the annotations or RNA-seq that those sites are ever used. So what these are essentially false positives that max-and-scan makes, but for example, they could be decoy splice sites that have a strong consensus motif, but have a silencer motif nearby that inhibits that site. And those are exactly the kinds of things that we want to learn. And so for the fourth group, we are essentially just scanning and picking random sites that will look very, very different from real splice sites. And for the next step, so the first step described the input to the model. So these are our targets. We actually need to quantify splicing, and the way we're doing it is, so we're looking at one donor at a time with multiple possible acceptors. And we're looking at the aligned GTEx reads. And you can imagine to get a naive estimate of the probability of each site being used, we can just count how many reads align to each acceptor. In practice, we use a slightly more complicated Bayesian bootstrap method that essentially allows us to estimate the standard deviation. And we're using that as an extra filter to eliminate low confidence events from our data set. So when we put all of this together, we find about 170,000 splicing events that we are putting in our data set. On average, each of these events has 1.2 annotated sites and 3.7 de novo sites. And to that, we are adding around 20 of these decoy sites and 60 of these randomized sites. So now we have all the input for our model. So let me show what our architecture looks like. Again, we have our conditional donor with multiple alternative acceptor sites. Each of these acceptor sites is being analyzed by a submodule of this network that we call a scoring network. And the scoring network outputs a scalar splice site strength score that is then normalized using a softmax layer, which then produces our predicted psi distribution that we can then compare to the true psi distribution. The trick to make this architecture work with any possible number of splice sites is that the weights between these scoring networks are shared, so we can replicate the scoring network as many times as we want. And also, the scoring network contains all of the model parameters, so that the number of model parameters doesn't change with the number of splice sites. So for the actual architecture we use in the scoring network, we tried multiple different architectures that you can read about in more detail in the proceedings. But the architecture that worked the best for us is when we use a series of convolutional layers at the bottom. So these are the layers that learn the features from scratch from given the sequence. Then we follow with multiple fully connected layers that integrate all these features. And then we use an LSTM, which is a recurrent type of neural network. And the LSTM connects one scoring network to its upstream and downstream neighbors. So these scoring networks can now exchange information directly. We train this architecture with cross-validation. What we do is we divide the genome into 175 contiguous regions and then assign these regions to 5 different folds. So all these performance numbers are obtained on held-out test data. On the left, you see the accuracy, which is the probability that the model picks out the strongest site out of all the alternatives. And on the right, you see the R-squared, which is the overall measure of how well the predicted psi distribution matches the true psi distribution. We use max and scan again here as our baseline by predicting the score for all splice sites and normalizing it with a softmax. Max and scan achieves about 35% accuracy and 8% R-squared. Our best model, which is the LSTM model, achieves 70% accuracy and about 60% R-squared. We experimented by adding a ResNet to the LSTM, but that essentially produces the same performance. And the other 2 models you can see here, the independent model is a model that lacks the LSTM, so it cannot communicate with neighboring scoring networks, which performs quite a bit worse. And the communication net, that's a simpler communication architecture where each convolutional layer can write into a shared buffer that is then shared with all the other scoring networks. And that gives a small advantage, but not as much as the LSTM. We're also stratifying the data set here. The blue bars are is the performance on all the events that we have. And the green bars is the performance on only those events where we have 2 or more annotated splice sites. So those events are quite a bit harder to predict, and you can see that all the models' performance suffer here. And in fact, you can even see that for max and scan, the R-squared on the alternatively spliced subset becomes negative, which means it performs worse than predicting a uniform distribution. So next, we don't only care about the predictive performance when we build splicing codes. We also want to know whether our splicing codes capture the underlying biology and replicate some of these regulatory elements that we already know about. And even though deep learning has a reputation as a little bit of a black box, there are some clever methods how we can look into this black box and understand what kind of features are being learned. So what we're doing here is we're following a technique from the DBind paper. And what we're doing is we're running a whole lot of test sequences through the network, and we're looking at the activations and only keeping those sequences that are achieving an activation above a certain threshold. Then we can align all the sequences and compute a PWM, which we can then visualize as a familiar sequence motif. We then take all these PWMs that we learn, and we use the TomTom algorithm to compare them to the regulatory elements that we already know. And so here we compared our motifs against the human splice site, acceptor, and donor consensus sequences, and you can see that COSSMO learns motifs that are almost indistinguishable from the ground truth. And we also, when we compare our motifs to the RNA compete database of RNA binding proteins, we find a lot of matches against known splicing enhancers and silencers. In particular, we found a lot of matches against SR family proteins, which are known splicing enhancers, and HNRMP proteins, which are known splicing silencers. Lastly, I just want to point you to our website that includes a web app where you can obtain interactive model predictions. We're also making our data set available because when you're building these splicing codes, a lot of the upfront work has to go into building a data set, so we're hoping that this makes it easier for other people to get started building these codes and implementing their own ideas. We're also making our code available on GitHub, and we're releasing pre-trained models that you can use as part of your own pipeline. So lastly, I just want to thank my colleagues at Deep Genomics and at the University of Toronto in the Fry Lab and the Morris Labs. So thanks a lot. I'm looking forward to your questions.
Now it works. We have time for questions. Yes, please.
Hans, great talk. So I'm wondering, what do you see as the hurdles to taking these sorts of models to be unbiased towards a prior splice site definition, to just throw huge span of genomic sequence at it and expect it to find splice sites out of that?
Yeah, I think generally nobody has solved that yet. So there are essentially two approaches to this problem, and one is our approach and the approach that most other people have taken before, which is to train on the natural genome, whether with or without available genotypes, and the other one is doing massive parallel reporter assays. They both have, so the problem with the MPRA is you're looking at a lot of random sequences in the context of just 2 exons. So you're limited in that way. I think so far nobody's really solved that.
Next question.
I have a quick question. I think there was another tool from your group, Spidex. What's the difference between this new tool and the other one?
Yeah, so it pretty much comes down to these objectives that are listed there. So Spidex only modeled cassette splicing and only binary events, and it was learned from a hand-engineered feature set, and it didn't include a background model.
If I may, I have a quick question here. Yep. Sorry. I really like the idea of using the output of an existing model to determine a interesting negative set, which you max and scan for. I was wondering, could you just use your approach in Bootstrap and train your model using an existing negative set and then see where your own model does make mistakes, which might be even more interesting and profound than max and scan, then include those and retrain? Is that something that makes sense, or is that maybe too slow, or why did you go for the max and scan approach?
So if you look at the proceedings, there is an additional figure that also looks at the types of errors that COSSMO makes. Yes, you could think about whether you can feed that back and have like one model build on the other. That would be interesting.
Okay.
Sorry.
Oh, one last question.
Yeah.
Thank you.
I don't think this is working. Just a quick question. Have you tried looking at the branch point usage at all when you're talking about the alternative splicing? Like, have you used it as an input?
Sorry, have I used what as an input?
The branch point information.
So the sequence window we use captures the branch point in most cases. So we look for the nucleotides upstream of the acceptor.
Okay, let's thank Hannes again for a great talk.